Good morning, ladies and gentlemen. Welcome you all to SIMPIC 2025 conference. It is truly a pleasure to have each and every one of you with us today.
It's not every day that we have the opportunity to gather in one place, surrounded by experts and peers, to explore ideas that could redefine how we approach science and medicine. This morning, we invite you to step into a space where knowledge meets curiosity and collaboration sparks innovation. Let's make the most of this opportunity to learn, share, and grow together.
My name is Chayanisa Pathanasiri-Mongkol.
And my name is Viranpathiranpruk. We are honored to serve as your Masters of Ceremonies for this morning's conference session. This session will run from 9:30 a.m. to 11:30 a.m. Each speaker will have approximately 30 minutes for their presentation. We encourage you to hold your questions until the designated Q&A time at the end of each talk.
Today we are privileged to welcome three distinguished speakers. First, Associate Professor Dr. Kasem Kulkeaw from the Department of Parasitology. Next, Associate Professor Dr. Iyarit Thaipisuttikul from the Department of Microbiology. And Dr. Punn Augsornworawat from the Department of Immunology. Please give a warm round of applause to our speakers.
Each of them will share their groundbreaking research and insights with us. Our first speaker, Associate Professor Dr. Kasem Kulkeaw, will present on the topic Unleashing the Power of Organoid Technology to Revolutionize the Study of Parasitic Infections.
Following that, Associate Professor Dr. Iyarit Thaipisuttikul will explore the topic Understanding the Microbiome, a new frontier in human health.
Finally, Dr. Punn Augsornworawat will discuss Understanding and Engineering Biology through Single-Cell Sequencing.
After each presentation, we encourage you to actively engage during the Q&A session. Your question and insight will enhance the discussion and deepen our understanding of this topic.
Once again, welcome to the SIMPIC 2025 conference. We hope this session will inspire collaboration, innovation, and a deeper exploration of biotechnologies, paving the way for the future of precision medicine.
Without further ado, let's begin. Please join me in giving a warm welcome to Associate Professor Dr. Kasem Kulkeaw, who will start us off with his presentation on Unleashing the Power of Organoid Technology to Revolutionize the Study of Parasitic Infection.
Okay ครับสวัสดีครับ Good morning everyone. Let me kick off my presentation with this slide. So as you may know that sometimes we cannot solve the problem with the old mindset. So today I'm going to open your mind to see about the organoid technologies, how this technology can address the problem that sometimes we face the challenge to solve that problem. So mainly I'm going to talk about how we can use organoid for parasitic infections. So let's get started. So as you may know that much of the knowledge comes from the study using the cell culture system as well as animal experiments, right? But not all of the knowledge can lead to the clinical applications, and that we call a fall in translational research. So the key question is that how we fill the gaps of this knowledge, and organoid can be an alternative ways that you can answer the gap or can fill the gap between scientific discovery and the clinical applications. So just briefly about myself, when I did my postdoc in Japan, so my research focused on what we call developmental biology. So we try to understand how stem cells generate the tissue and organs by using mouse and zebrafish embryos, and I used hundreds of embryo in order to understand this process. So if you're familiar with the animal experiment, you have to answer this question when the committee asks you whether how many embryo you're going to use and what method that you will use with the embryo, any pain and discomfort that generated during the experiment, and also why do you need to use the embryo for this experiment, right? And that is according to the rule that we call 3R. Have ever you heard about this? reductions, refinement, and what is the third one? Sorry? Replacement. Yeah, you got the point. We have special award, right? Oh no, sorry for that. Okay, replacement, and that is the keyword for today. Organoid can be an alternative model to replace the animals, but not now. It's still on the research and improvements, and that we will discuss about as well. Okay, so that is the title of my talk, and to show you that how organoid technology can change the way that we understand the disease mechanisms, especially parasitic infections. So our goal in our lab is that we try to develop the alternative model for understanding human diseases, especially infections caused by the parasite. So I divide into three parts. First, I give you some basics about organoid technology, and then I will give you some examples of the applications, but not in detail because we have only 30 minutes. And the room for improvement, that's important because it's the rooms that you can sit in and do more research about how to improve the organoids. Okay, so for the first part, so we have to appreciate the previous attempts from scientists more than centuries ago that they try to understand when you dissociate the cells, dissociate the organism into single cell, they can reaggregate to form the tissues as well as the whole organism. So this is the study in sponge and chick embryo, and that leads to the discovery of stem cell, เซลล์ต้นกำเนิด in Thai, sorry for that. So stem cell, and stem cell is a keyword that we use that stem cell to generate the organoid. I will touch that a bit about what is the stem cell and what kind of stem cell that we can use. I don't know where is the timer. Okay. So let's have a look at the very important study, the pioneer work from Professor Han Clever. So he first demonstrated that how you culture the stem cell from the crypt of the small intestine. So what he did is that, as you know, in the crypt of the intestinal organs, you can see the stem cell in this area. And this is the villi that when stem cell need to replace the damaged cell. So what happened here? So he isolated the stem cell from the crypt and culture by embedding cell in the 3D structure, in the scaffold we call the gel. And the cells undergo cell proliferations, here. And then they start to protrude the dome-shaped structures. Can you guess this dome-shaped structure equivalent to the villi or the crypt? Villi or crypt? Villi. Villi also? Okay. The outside area, this is the extracellular matrix. So the dome shape that protrudes in that extracellular matrix regions is the crypt. So you can find the stem cell here in this structure. So yeah, this is a very pioneer work that to show how we can get the organoid from adult stem cell. So the second keyword is adult stem cell. So the key character of adult stem cell is that so the organoid consists of, thank you so much, that's very useful, very helpful. So the organoid have more than one cell type, and also they're able to cell organize into the structures that's similar to the origin of the tissues, as well as the functions. This is the three characteristics of organoid. If you generate organoid in the lab, you have to show that first it contains multiple cell types. First, they have the structure resembling the original tissue, and third, they function similar as well. Okay, this is the key characteristic of the organoid. And this is the source of the organoid. You can generate the organoid from pluripotent stem cell, which is the cells in the inner cell mass of the blastocyst. And also, you can generate organoid from the iPS cells, induced pluripotent stem cell. This cell comes from somatic cell that you turn it to be the pluripotent stage. And also, you can isolate adult stem cell that reside in the tissue here. Mostly, this multipotent stem cell can work or responsible for maintaining and repairing the damaged cells. So this is a key source of the organoid formations. So the question is that what is the difference between 2D and 3D culture, right? Have ever you asked these questions? So normally people, when they grow the cell in the 2D cultures, the cell adhere on the surface as multi-layers. They expose to the gas and oxygens, or gas and nutrients in the linear manners. And that allows, in the monolayer, the cell interact with adjacent cells, just one direction. But when you grow cell into 3D structure, so you embed the cell in the semi-solid structure, so-called gel, for example. And this cell undergoes cell proliferations and form multi-layers. So when they form multi-layers in the gel, so the way that they expose to the gas and nutrients also not in the linear form. It depends on the depth of the locations from the liquid medium. So here the culture medium outside, and this allows cell to interact to the adjacent cell and all directions. And cell also have what we call cell polarizations. So this makes the cell different, but this is just simple cartoon to explain that. But let's have a look at some of the study to show that what is the difference between 2D and 3D cultures. So in this study, they used the iPS cell, pluripotent stem cell, and they dry this cell to become the liver bud. Liver budding is the early formations of the liver in the embryo. So in this study, they used the iPS cell to generate the mature hepatocyte, and they correct this cell. It's called hepatic endoderm. And they co-culture with the mesenchymal cells and endothelial cell, and that allowed the cell to form the 3D structure, as you can see from these pictures here on the top of the gel. So when they check the single cell analysis, and that Dr. Punn will cover about single cell technology, single cell RNA sequencing technology. So you can see the divergent point here between the 2D and 3D cultures. And both directions lead to the cell that express albumin, which is the indicator of albumin, hepatocyte, right? So can you guess which one is close or similar to the hepatocyte in the liver? The 2D or 3D? 2 or 3? You can show your hands. 3. Yeah, I didn't show the data, but based on transcription, the 3D culture method allows cells to become more similar to the hepatocyte in the livers. Okay, that's just brief introductions about what is the organoid. I show you the pioneer work as the root of this technology, and also key characteristics of the organoid, as well as the difference between 2D and 3D cultures. And then I will move on to the second part about the applications. Okay, what is the application? So I will start from the head to the bottom to show you some example. Some of them from our study. So start from amoebic encephalitis caused by free-living amoeba. And then we will dig into the liver to see the liver stage malaria. And then we go to the small and large intestine. Brain-eating amoeba. So our lab, we focus on this strain of free-living amoeba we call Balamutia mandilaris. So this is the this first discovered in the mandrill baboon in San Diego Zoo, and you can find this amoeba in fresh water and soil as well. This is counting down. Okay, 9 minutes. So when you get the infections through the skin wound or cuts, so it causes the lesions we call granulomatous amoebic encephalitis, as shown here. And you can see the necrotic cell, hemorrhage, and inflammation. So the mortality rate is very high, more than 90%. There is no drug, no specific drug, and also very difficult to diagnosis of this disease. So this is one of the very rare disease, and very difficult to get the tissue sample to understand about the pathogenesis of the disease. So that's why we generate cerebral organoid to study this amoeba. So first, we isolate amoeba from the clinical sample of the 4-year-old Thai girl who died from this disease. And as you can see here, you can see the trophozoite stage, which is very active in terms of metabolism at the cyst stage. And we use the 3D imaging to see how trophozoite look like. And then our staff generates cerebral organoid from iPS cell, which is the pluripotent stem cell. Following the previous protocol, And we published this work already in the PLOS Neglected Tropical Disease in last years. So this is what it looked like when we talk about cerebral organoid. You can see the different stage of the cells from the iPS cell to the mature cerebral organoid. And we, as I mentioned, we need to make sure that it contains multiple cell type, right? So we check this. It contains mature neurons, what else? Radial glial cells, astrocytes, and oligodendrocytes, as shown here. And also it functions in terms of calcium influx and efflux. So we co-culture this organoid, just simple experiment, right? So what happened is that, please have a look here. This is the amoeba, asterisk here. So it penetrates, invade into the cerebral organoid, as shown here. And you can see the damage. This is normal neuron cells and damaged cells. We also found that we could detect the brain trauma biomarker from this organoid. It means that there is damage of the neurons. And then the key point of this experiment is that we try to use organoid to replace the animal or even the human brain tissue. So let me introduce you about drug repurposing that lead to the discovery of nitoxurin. So the Lori, so 4 minutes left, right? 14, oh sorry. So Rory screened more than 2,000 clinically approved compounds and come up with the nitoxurin. Nitoxurin is the antibiotics that used for treatment of urinary tract infections. And this group also showed that in the presence of the drug, nitoxurin able to protect the brain. They use the brain tissue expand culture. You can see here. This is without the drug, and this is with the drug. You can see that it can inhibit the cell damage caused by amoeba. And that leads to the clinical applications. There's the one case that they show curative treatment. The patients can recover from the infections. Very fascinating about drug repurposing. And we try to examine whether instead of the brain tissue explant culture. This means you just make biopsy of the brain tissue and then put into the culture, right? So we ask whether we can use cerebral organoid to replace this tissue or not, because it's very difficult to access. So here, we first test whether the amoeba that we isolate are susceptible to the nitoxurin or not. This is the IC50 curve. You can see this from this value that quite similar to previous report. And then we perform similar experiment, but the difficulty is that it's very difficult to assess whether the drug efficacy work or not. So what we did is that we just simply counting the amoeba in the organoid, comparing to the host number of host cells in the sections, and you can see the reductions of the amoeba inside the cerebral organoid. For further information, you can read our paper. Okay, that is the brain. So now we move on to the liver stage. For the later part, just briefly about organoid applications. Liver stage malaria. So this is what it looks like when we generate the hepatic organoid from pluripotent stem cell. On the left-hand side, you can see the snapshot of the organoid image. You can see the spherical shape here. And on the right-hand side, oh, sorry, which one? Left and right, yeah, correct, right? On the right-hand side, you can see the video of the spheroid. You can see the layers of the cells, and they form like a ball with the space at the middle of the structures. So when we follow the paper that generate liver bud, and we did the same thing by generate the cells and mix them together and put on the top of the gel here, and upper phase is the liquid medium. So the cell start to form aggregations. And we also characterize that this contains the albumin secreting cells similar to the liver. So next, we add the sporozoid of the P. vivax into the culture system. So in order to ensure that this organoid, liver organoid, allow complete schizogony of this parasite, we follow the schizon stage as well as the morozoid. So you can see this is the marker of Parasitophorus vacuole inside the cells, as well as we prove that the release of morozoid can infect into the human reticulocyte, as shown on the right panel. So However, there are limitations in this experiment. First, very low infection rate that we found, and also when we use cryopreserved sporozoite, it's very difficult to get very high infections rate. And also, there is the barrier, as you can see, that because the compact structures of the organoid that might be difficult for sporozoite to penetrate into the structures, นะครับ and also it requires human reticulocyte that very difficult to isolate from the blood, นะครับ Okay, that is a second example, and now we move to the third and the fourth example, นะครับ this is in this experiment they generate mouse intestinal organoid from the adult stem cell in the crib, and they demonstrate that how this mouse intestinal organoid can support the complete development of Cryptosporidium parvum, นะครับ So this is the cartoon that shows the life cycle of Cryptosporidium parvum that attach on the surface of the cells. So what they found here, first they found that the trophozoite can attach on the intestinal epithelial cells, and then they're able to develop asexually into two types of the neuron according to number of sporozoites inside, นะครับ and also they're able to generate the sexual stage we call macrogamon and microgamon, and that these two stages able to make a fertilization and generate what we call oocyst, and that infectious to the mouse, นะครับ so this demonstrate that Cryptosporidium parvum can completely develop sexually and asexually in this organoid, but they still use mouse intestinal organoid, so cannot overcome the replacement as I mentioned, นะครับ I think no study report about human intestinal organoid. That one of the room that we can further investigate. And about large intestine, นะครับ so this study generate mouse cecal organoid. So this is the intestinal nematode called Ticulis muris. The key question for this nematode is that how the larva develop inside the epithelium layers of the large intestine. That is the key question that this study clearly show that there is the early tunnel formation that the larva can form inside the cell here. Inside the cell, you can see the worm, this is the whole cell, and then they start to grow up in that layer, and then that's why it might be answer the question of how it penetrates into the layer of the large epithelial cell, นะครับ you can further read that paper. They not only demonstrate the development of the larva, they also study how the host cell respond to these infections. Okay, that's the overall. So I show you that we use cerebral organoid to show you that there is a cell damage, and also we prove that cerebral organoid can use for drug test. And for the liver stage, malaria is still need further experiment to increase the infection rate of this liver organoid. And for cryptosporidiosis, mouse intestinal organoid can prove that how the parasite completely develop in that system, as well as the whipworm infection. You can see very early larva development in that the cecal organoid. Okay, so the last part of my talk, so I have about 9 minutes, right? Okay, good. So I think we should spend more time to discuss about this, นะครับ room for improvement. So I just sum up here. Before we see how we can improve organoid technology, let me move to that part. So the goal of organoid technology is that we try to recapitulate our tissue, right? So this is the organoid. So if we compare with the conventional 2D cell cultures, the key difference is that the complexity of the model. The organoid have more cell type, and also they can structures that similar to the tissue of the origins. But the problem of organoid is that the organoid is static. Static means it's just lack of the dynamic flow of the fluidic system. That's why organ on chip turn to be the promising model that you can connect tissue or different cell type together via the vessel-like structure นะครับ So here, this is the room for improvement. I sum up here. So first thing is that you can see the compact structure of organoid is very difficult to use the light microscopy or inverted microscopy, right? So you have to make a section of the tissue similar to the pathological examinations. So cell imaging still challenging. Second thing is that if we have organoid lines, it's very convenient to culture the cells without stem cell isolation and cultivations. Organoid lines, as same as cell lines. And what if we can make a cryopreservation of this organoid? So it's like a on shelf. So when you want organoid, you just thaw the cells and then culture in the dish. Moreover, the current organoid method still relies on the animal product, so we cannot overcome the replacement of animal นะครับ So many people try to find the alternative materials that can replace the animal product, as well as the cost of the method still very high. So if we find a cheaper way, that would be nice. For the tailor-made, what does it mean? So if you use iPS cell, you can knock out or knock down the genes or the cell that you want to prove that this is play a very important role in the pathogenesis of the parasite. You can prove that in this system นะครับ But it might be difficult for adult stem cell because adult stem cell are very difficult to grow, to expand in the dish, comparing to pluripotent stem cell นะครับ For parasitic infections, what we can improve first, you see this organoid lack the vasculatures and immune cells. As you know that these two systems also require for pathogenesis of some parasite infection, right? And the second thing that you can do is that you can improve is that because the compact structure of the organoid that have the lumen inside is very difficult to demonstrate how the intestinal nematode infect in this system because they inhabit in the luminal organ นะครับ Because it's a compact structure, it's like a ball. How the nematode can penetrate into that structure, it's quite challenging นะครับ Okay, I sum up here. So what I show you just basics of the organoid biology as well as how we can apply for parasitic infections, and there are many rooms that you guys can improve นะครับ Sum up here, what is the organoid? Is the miniaturized form of the tissues, right? And they have same structure and functions as same as the tissue of origin. You can generate from stem cell that we call adult stem cell, which is the multipotent stem cell. So they can self-organize themselves into the structure that's similar to the tissue of the origin, as same as they're able to maintain themselves as the tissue in the dish because they still have the stem cell inside the organ นะครับ This is the liver organoid that we first generate in our lab. Okay, just allow me advertise about our research. So we have some project that related to the malaria research. As you can see that now we still rely on animal experiment, even mosquito. We try to develop all-in-one system to use the organoid technology. So start from the liver organoid here. And also one of the key questions for PYVAC is about reticulocyte infection. So we try to generate reticulocyte from the cells. And also midgut organoid from the mosquito. This is one that might replace the mosquito facilities as well. Okay, ครับ So I would like to thank our collaborators and the grant agencies นะครับ that support this work, and also thank to our lab members นะครับ and our Thai collaborator that support this work. If you have any questions, you can contact me by this email. So to protect personal information, I select these pictures to hide their face นะครับ Okay, I'm happy to take all questions regarding to the organoid technology นะครับ Thank you so much.
Thank you, Associate Professor Dr. Kasem Kulkeaw, for your enlightening presentation. Now we have some time for questions. Feel free to raise your hand and we'll bring the microphone to you.
Sure.
Thank you very much. Ajahn, I'm just wondering, this organoid technology is very fascinating. I'm wondering, like, if the cell will keep growing, or do they have the same property like the organ, like at some point they will stop growing? And if they do keep growing, like, do we subculture them and then make another mini organoid, or like?
Yeah, that is a good thing. That is a thing that many researchers try to establish organoid as the cell lines, so you can continuously grow the organoid by just simply subculture. Maybe you guys familiar with the cell culture, right? You can subculture cell for the long-term cultures, and the problem is that cells going to turn into what we call senescent, and that will stop cell will stop proliferate, and that's very difficult to subculture. But believe me, the scientists try to overcome that limitation, so that you can have continuous culture or limitless supply of this organoid for your research. Yep. Did I answer? Okay, thank you. นักเรียนก็ถามได้นะครับ
Are there any further questions?
Yeah, from กล้องครับกล้อง I know you. The medical student who are very active in research.
Not that much. สวัสดีครับก็ I have a questions about when you talk about the use of supplement from animals, like you want to try to reduce the use of it. I mean, like fetal bovine serum or something like that. I want to know, do you have any idea about the use of another supplement from instead of animals?
Okay, that's a good question as well. So, as I mentioned, one of the methods to generate organoid is to embed the cell in the semi-solid, and that we call scaffold. Scaffold can be any kind of gel or polymer, not only from animal product, for example, from the plant, like a cellulose or methyl cellulose, and that make a scaffold. But the key components are extracellular matrix, right? For example, collagens. You need to add the collagen and growth factors in that structure. So to answer your question, what we can improve is that instead of the whole extracellular matrix, some people try to make a peptides and cross-link them together to form the adhesive molecule, so that cell can attach on and start to proliferate. So you don't need to use animal product. You just use the peptide that cross-link together. And one thing, in addition to growth factor, well, now the growth factor can be produced from the cell lines. We don't need to use animal. So now you can see in the future, totally animal-free system. No? Hopefully we can see that in the future. ครับเชิญครับ MC ก็ถามได้นะครับ
So I have a question, like whether the organoid has a defensive mechanism the same as the human body, because like, for example, you explain about the P. vivax, like, so in the human body, I believe that when P. vivax tries to like infect the hepatocyte, there might be a defensive mechanism from the body. So does the organoid also replicate that? Yes. Some studies show that once the cell get infection, they have the what we call innate response, and that also observed in the liver model as well. Not only P. vivax, the hepatitis C virus infection, interferon mechanism also switch on. For the P. vivax, people realize that there is the response in terms of cholesterol synthesis. We don't know why that changed in the liver model, but that happened also in the organoid and also in the human body. That's good thing that can tell us that whether the organoid can fully recapitulate our body or not, right? Thank you.
Okay, I have a question too.
Thank you. That would be great.
Can you please talk a little bit more about the implications of reducing animal testing with this technology? Or removing, no longer needing animal testing.
Not only the cost of the organoid generation is also applicable for regenerative medicine. Actually, organoid is supposed to be a way that we can regenerate the tissue when there is the damage or you need to replace the damaged tissue. And if it contains animal product, that will be the problem for the allergic people or cause any side effect. That the thing that I didn't mention. Thank you for that. โอเคนะครับ หมดเวลายัง
The floor is still open for any questions, so please use this opportunity to ask our professor. One more question.
ครับ
Sorry, I'm just wondering if you co-culture the cell together and form, like, for example, the liver bud or the intestinal, do the cell arrange into the arrangement similar to the tissue, or they just randomly placed in the bud?
I see. Yeah. For the liver bud from iPS cell that I just showed you, surprisingly, the cell can organize into the structure that resemble the what we call robu, the microstructure of the liver. You can see a cluster of the hepatoblast surrounding by endothelial cell. But that one of the things is that if you generate the organoid from iPS cells, the cell, the cluster or the organoid that you got is similar to the fetus, the fetal stage. So many people try to overcome that, try to get more maturity of the organoid technology. That's one thing that I didn't mention here. But for some organ, like a cerebral organoid, sometimes we get the randomly organizations of the tissue in the brain. But yeah, many people try to optimize the protocol so that they can get the microstructure similar to the brain or the liver. That's one thing that's very challenging. Thank you.
Are there any other questions? Okay, if not, I would like to thank you all for your interest and inquisitiveness.
Thank you so much, ครับ
Once again, thank you to Professor Kasem Kulkeaw. Up next, please join me in welcoming our next speaker, Associate Professor Dr. Iyarit Thaipisuttikul, who will be presenting on Understanding the Microbiome, a new frontier in human health.
Hello. Okay, so good morning, everybody. So thank you for joining us on Sunday morning. All right. So my topic is about microbiomes. All right. So I believe that everybody heard the term microbiome very well. All right. It's a very hot topic in the past, about a decade before. All right. So actually, it's not just mean that. Until this moment, all right, the concept of microbiome are not quite actually new. All right, so there has been a very extensive studies for quite a while, but I'm gonna tell you that actually even with the extensive study, like now, we are just at the beginnings to understand what does it mean. All right, but there was still a lot of works, right, before we can apply this knowledge in medicines. So in terms of microbiomes, all right, so you can see that actually this is kind of like the concept of normal flora that we have been known for a very long time, like centuries ago, all right. But for the microbiome itself, all right, so the term has been generated only about 20 years ago, around that. And just to orientate everybody to get on the same page. So when we talk about microbiota, all right, microbiota, actually this is a term that should replace the term normal flora, which is the old term. All right, nowadays we should call it microbiota. When we talk about microbiota, so we talk about microorganisms which have the association with the habitats. All right, If I have E. coli, and I just say E. coli alone, it's just E. coli. But when I talk E. coli in association with gut, right, that E. coli is a microbiota. All right. So in term, in this, with these definitions, actually all microbes on Earth have habitats, which means all microbes on Earth are microbiota. It's depending on what habitat that we are focusing on, whether it's in the gut of human, the air, in the soil, in the water. So actually everywhere have microbiotas. But when we talk about microbiomes, microbiome is mean you talk about the totalities of microbes. It's not just a single individual microbe, but we're talking about the communities of microbes. All right. But the originalities of the term microbiome are quite interesting because it has been believed that it was coined by Joshua Lederberg in 2001, all right, based on the omics term, which means we're talking about the collective genomes of the microbial communities. But turned out that the term microbiome had been used long before that, which means the ecosystem of the microbes, which means that for the term microbiome now, it's kind of like the combination of both. We're talking about the ecosystem of microbes plus their collective genomes, right? Because right now, the method that we use to study microbiome is genomic based, meaning that once we study microbiome, we're gonna know their genome content as a result of the studies. And microbiomes, actually the whole term includes all type of microbes, bacteria, virus, fungi, eukaryotic microbes. But because the way that we study, the method to study for bacteria is now the most well established and very stable, and there's many tools that we can use for study bacteria. In terms of, but instead for virus, fungi is more difficult. Eukaryotic microbiota is the most difficult one to study, which means that if you look into publication and you see the term microbiomes, right, most of them mean bacteria. So in that case, virus, if somebody study viral microbiomes, so they like to use the specific term called viromes. Or fungi, they use the term microbiomes. All right. So actually, in publication, I saw some researchers try to use the term bacteriome to specify bacterial microbiome, but I believe that term are not so popular. So nowadays, every time you see the term microbiome, so most of the time it's mean bacteria. But for microbiomes, all right, so we talk about the ecosystems of microbes, all right? But as we study more of it, it's not just the ecosystem of only microbes, right? But they form the ecosystem with their habitats, meaning that part of the microbiomes, for example, in human, like in gut, in our gut. So those ecosystems, including our gut, and they have association, they have the communication with our own cells. So, and that leads to the many implications in terms of medicines, like, so they can promote health or promote disease in some cases, and that we have to stay tuned for a new knowledge that coming up each day. Okay, this is kind of like interesting fact about microbiomes. I'm not gonna talk all of it, but there are many very interesting numbers. You might heard some of them, like, in total human bodies, we have the number of microbial cells, right, or microbial entities, about 1.3 or 1.4 more than our own cells, all right? Which means that when you see human, like you or me, when you see human walk on the street, right? Half of them are human, half of them are microbes. All right. Okay. And when you look at the compositions of microbes, in this case, it's bacteria, from several organs, like anterior nares, like buccal mucosas, all right, tongues, stools, vaginas, all right. You can see a different type and compositions of microbes, all right. So this is a stack bar chart, all right. One stack is kind of small. The one bar chart here is represent one human, all right. And you you have several humans lined up together, and you're gonna see a chart like this. You can see that there's so much variation among individuals, human individuals. For example, in stools, probably the most specimen that we study in stool. This is kind of typical microbiota profile at phylum levels in human stools, right? So every time we did the research on human microbiome, on the stool microbiomes, we always see this picture, like you're gonna see the line from one side to the other side like this. This is a line that divides the phylum Firmicutes and Bacteroides. All right, so in human population, there will be variation in the proportion of these two phylums, right? And for the rest, there are another minor phylums. This picture is kind of like very interesting in terms of if you look at several organs like this, you see a lot of variations, all right? Except one organ that unite all human on Earth, all right? All human on Earth for one organ have exactly the same microbiota profile, which is vagina, all right? So all human female on Earth, we have the same, all right? The same microbiota profile, which is dominated by lactobacillus. Okay, so in case of gut microbiomes, right, because the our human intestine, GI tract, this is an organ that hosts most numbers and type of microbiota in humans, and they involved in several physiologic processes. All right, so they have they have a lot of function involving human health, like help the digestion, right, immune system modulations, nutrient small molecule synthesis, include and protection from the pathogens. And this is the same thing that kind of the same picture that I showed you before, but this is by our own study. This is our preliminary study, like several years before, just to look at the gut microbiome profile in Thai population. But the the sample size is kind of small, but you can still see the classic pictures of the gut microbiomes, that you have the several different proportion of Firmicutes and Bacteroides. All right. So in human gut microbiome, the most common compositions of human gut microbiome will involve 4 phylums like this: Firmicutes, Bacteroides, Actinobacteria, and Proteobacteria. All right. But because we are microbiologists, so some of you probably know that the name has changed a couple of years ago. Right, Firmicutes to Bacillota, Bacteroides to Bacteroidota, and so on. All right, but I believe that these names are not so popular. People still call it Firmicutes and Bacteroides. All right, but because of the name change, it's causing some confusion in the field. But usually gut microbiomes in humans are quite stable. All right, so they are not kind of stay the same, but they're kind of dynamics based on the influence from the environment, like diet or xenobiotics and so on. But they always swing back to the baseline of each people. Right? But the factor that influencing the compositions of human microbiomes, all right, I think the one that have the very serious effect and the most effect is the antibiotic use. Right? So once you take antibiotics, it will drastically change the composition of the gut microbiomes. All right, and it takes some time for the gut microbiome to reverse back to normal. The other one is diets. All right, diets. It depends on the type of diets that we eat. Right, more fibers or more meat-based, or more fat, more lipid-based. But the diet kind of change change type and can change the composition of gut microbiome, but not so drastic like in the same levels as the antibiotics. And the another things, environmental, lifestyle, those are the kind of minor factor that can influence the composition of gut microbiomes. Pollutions, all right, stress, exercise, and so on. Okay, in terms of method, all right, in terms of method, nowadays we use the sequencing-based method in order to study microbiomes. So most of you probably know the terms, all right? So we can either use to do the amplicon sequencing using the marker genes, which is the most popular one is to use the 16S rDNA, or we can do the shotgun metagenomics. We can get all the informations, but of course the cost is very high nowadays. For 16S RNA, all right, so there are new technologies that could have implication in the future, because at this moment, right, the most common method for us when we do 16S sequencing is to use short read technologies like Illumina's. We can take only a very short region of the 16S. All right, but nowadays we have long read technology that able to do the full length 16S, which is gonna get you more information, all right, and can render more resolution. Sometimes you can look into even the strength of the bacteria. All right, but I believe that nowadays during the transition, and people try to apply the wrong read in order to do a 16S. But then again, there's a dilemma about long read is that, if you can do a long read 16S, why don't you use long read to do shotgun, right? And you get more information. Well, that we have to leave that on bioinformaticists to make sure that which method is the best one for us. Okay, so if you want to conduct a microbiome research, all right, so basically the basic one is to compare populations. It's kind of like clinical research. All right, so microbiome is just one factor that you're gonna see either or not it difference among the group that you're gonna compare. So in general, right, so when you conduct microbiome research, start with study design, sample population, sample collection, sample prep, sequencing, and analysis, right? Sample design is the same as a clinical research. You have to design exclusion criteria, inclusion criteria, sample size, and so on. And then sample collections. I want to say that study design is the most important one. It is the same concept as a clinical research. You need to have a good study design in order to make sense of everything, all the results that you're going to get later on. Sample collection, right? So this is the way how you collect the samples, right? I want to say that sample collection is probably the part that most people overlook because it sounds simple, but in real life it's not simple, right? And especially for microbiome research, because the microbiome, the method to analyze microbiome are very biased against the method, right? If you use different method for collecting samples, especially from different subjects, it's going to give different kind of result and bias or your interpretations, which means that sample collection needs to be standardized and have to be conducted in the same way for everyone's in the studies. And then when the sample reach the lab, we prep the samples, extract DNA, and then do sequencing. And then analysis using the bioinformatics software, all right? So you can see that the whole thing here, this is make the microbiome research multidisciplinary, all right? So it involves clinical studies, all right? Logistic sample collection, the wet lab, DNA extraction, and the computer works, right? Which means that in real life, you probably cannot get one researcher that do best in everything. which means, it's better to get like collaboration from, our different experts in each field. You might need rely on doctors who can help you get the patients or the cohorts, all right? You probably need to rely on bioinformaticists to help you analyze it. So when you look into the PubMed, looking into the theme of microbiome research, right, you can see that they are most of the time fall into these three categories. First one is to study the basic biology of microbiomes. Right? For example, you take people with different kind of diseases and compare the microbiome composition among them. Second one is the applied studies of the microbiomes. How going to use that to make interventions or to make a test or diagnostics or whatsoever. So which we're going to talk about it later on. And the third one is more about the R&D to develop the software tools and system, the pipeline, and the resource, and so on. All right? Because now we are more like in the process, in the stage of collecting data. All right? Most of the study are in the first group here and in the second groups, just to how to we're gonna make sense of all this data. Okay, so for example, if you take a group of patients, for example, like you have a group of patients with normal weight and the other group of patients with obesities, all right, BMI over 30, for example, and you compare the gut microbiomes, all right? So you can see something like this, all right? So you can see, okay, I'm not sure you can see that this is control, all right, this is obesities. All right. So, not using any scientific knowledge at all. You just use your eyes. Are this group and this group different? Just use your eyes. It looks similar, right? But if you try to look more, all right, just make it very simple. Right? You probably can observe that for the obesities, right? They have lesser colors. They are, in control. They have more colors, right? The more color it is, meaning that they have more type of phylums, as shown here. All right. So this is one typical findings of obesity, which is they have lower diversities. Right? But I just show you that, okay, this is just to make kind of, just to speculate what should be different. But actually, everything has to be calculated statistically. And most of the time, this is kind of classic observation of the gut microbiome profile in obesity. For example, they have more firmicutes, they have more gram-negative, they have less diversity, and so on. All right. So, not just obesity, right? So there are so many diseases that people at this moment study and see the difference in microbiome compositions between healthy individuals and the diseased individuals. But the main question is that, what does it mean? So what? If I see more E. coli in obesity people, and what does it mean? All right, and how are we gonna make sense from that, or how are we gonna use that? All right. For example, if you try to interpret this result, all right, is microbiome profile difference that you see between normal weight person and healthy and obesity person is a cause or effect? All right. For example, at the beginnings, right, these two groups, healthy individuals, might have the same type of microbiome compositions. But because of when people get obese, the physiology in their body change, and they involve like environment in the gut, and then make the different kind of suitable environment for the microbiome, and then microbiome start to change and become different. All right. If that's true, it means that microbiome difference in obesity is an effect of the obesities, effect of obesities. Or in the opposite way, all right, at beginnings, these two group of people, right, might have different microbiome composition at the beginnings. And because of that microbiomes in the obesity, that lead to the obesity in that patients. Which is, in that case, that's gonna mean that that gut microbiome is the cause of obesity. All right? And if that's true, if it is true that microbiome is the cause of obesity, that changes everything. All right? In the past, right, when we talk about what is the cause of obesity, right, or several factors, genetics, behaviors, environment, and so on. Nowadays, if this is true, now we're going to have a microbial cause of obesity. Right? Okay. This is a landmark experiment done in 2013. Very famous. You might heard about it. All right, so the researchers take the stools from obese and lean twin. Right? This is just to remove the confounding factor about the genetics. Right? But they have twins, one obese, one lean. They take the stools from both, all right, and transplant that microbiota into germ-free mice. All right. At this stage, these germ-free mice are the same. So now that these germ-free mice that get microbiotas from the obese one become obese mice. The one that get microbiotas from the lean one become lean mice. What does it mean? What does it mean? Yeah, and not just the obesity. Nowadays, they have kind of the same kind of study like this with several other diseases. Like for example, the people take stools from the people with depressions and transplant into germ-free mice, and they got a depressed mice, for example. Which is mean that microbiota actually behave like a cause of the disease. Right? Probably not a sole cause, 100% cause, but they for sure participate in the pathogenesis of the disease. Right? You can see that just by this fact, that changes perspective of human disease. Right? Because because we usually think about human disease, especially for NCD, non-communicable disease, are caused by genetics, environment, and so on. Now, some of them are caused by gut microbiota, all right? And that, nowadays, they have several diseases, all right, that involving microbiotas, especially neurological disease that we call gut-brain axis. And nowadays, there are many more, gut lung axis, gut liver axis, gut joint axis, gut skin axis, and so on. And this is a landscape of, once we know what we could use that for in medicines, we can use the gut microbiome profile as biomarkers. For example, predicting the disease actually for every stage of human life. Right? Disease prediction in pregnancy, infant health. In the infant, predicting the disease, tracking gut colonization, or microbiomics. The term microbiomic is that some of the drugs that we take will be metabolized by gut microbiomes, which means that if you have several individuals have different gut microbiome, then you could have different effect by the drug, which means that sometimes you may need to see whatever gut microbiome profile is before deciding which drug should be prescribed. In adults, right, predicting the disease, and in the disease in patient who already get the disease, all right, to diagnose the disease and to make a prognosis, to do a precision medicine, all right. In terms of interventions, right, now we know that if the disease are caused by microbiota, which means that if we can manipulate or change microbiota, whether or not we could treat the disease, or we could treat the disease or prevent the disease. All right. You can imagine that in the future, and it's not true now. It's not true now. In the future, you can imagine it is possible that if we can treat people with obesity or diabetic or whatever disease by drinking yogurt with specified microbiota, all right? But that's the long way to go. Okay, so this is the intervention now at this moment at the current state here. So now that what intervention which people can do with microbiota now, right? So we can do fecal microbiota transplant. We can use microbiota to predicting the diet interventions. We can use fermented food with have a natural natural microbes, functional microbe in it. We can use prebiotic, probiotics, phage therapy, or postbiotic, which is the microbiota metabolites. Okay, in terms of probiotics, this is different because probiotic is a term that has been used for a very long time ago, but most of the probiotics that we use now are traditionals. If you have a chance, go into convenience store like 7-Eleven, look into the each of the yogurts or the food that have the probiotic. You probably see the kind of the same names all over and over again. Those are traditional probiotics that have been used in the history for a very long time, like Bifidobacterium, Lactobacillus, Saccharomyces. This is a probiotic that has been isolated in the food, in human food, and they are certified to use it as a probiotics. But but when you ask probiotic works or not, in your opinion, everybody have the different opinion. When you say that probiotic can promote your health, is it true? Or it's just marketing plan? All right, so if you look into our research, our metadata, our meta-analysis, you're going to see that probiotics have no evidence that probiotic can provide advantage. All right, but they are not harm as well, meaning that they are kind of neutral. All right, so but you can think that if we want a probiotic to treat our conditions, all right, to treat our condition, treat the disease, all right, and we know that each of us have different gut microbiomes. Is it possible that we're gonna take only one type of microbe and fix everybody's conditions, right? Even though we are different, this is why probiotic might not be. If we can just keep following, using the same type of probiotics. Right? In this case, there are a new type of probiotics called next generation probiotics. Right? This is from microbiota or microbiome research. If you take healthy control, disease control, and see the difference, and you see that one microbiota is more predominant in the healthy person, which means that microbiota might be a good one, have a protective effect and so on, and choose that one to make a probiotic. This is a new one. For example, Akkermansia, right? Bacteroides, some Bacteroides species, Faecalibacterium, and so on. All right. That's a new one that has been developed now. Okay. Even with all the knowledge that I just provide you, right? But in the future there will be still a lot of challenges. For example, we don't have what is mean for healthy gut microbiomes, which means we don't have standard, we don't have cutoff. If you take your own gut microbiome and see the composition, we cannot tell you that this is normal or not, right? Because we don't have cutoff. And because gut microbiome have so much variability among individuals and dynamically change depending on genetic diets and environment and lifestyle, this is very difficult to assign what is normal. All right. And you can see this is the same picture. You can see that actually everybody is in gut microbiomes in several individuals are different. All right. But if you look at here, it's second panel here. So actually I haven't shown you at that time. This is the same samples from each individual, but they look into functional genes instead of the species. All right. You can see that even though species among individuals are different, but their function are the same, which means that it might be It not just that you can look into the composition by looking at the species or genus, right? But the real thing is that you might have to look into their functions. Some microbes, even though they are different species, but they have the same function depending on their conserved gene in the genomes. Okay, so this is one example from a study last year. This is a very good study. I would recommend you to read it. This is from China that they identified the one microbe called Megamonas that associate with obesity in Chinese populations. And they prove everything in the labs very elegantly, looking at the mice and show the pathway and show the effect of this Megamonas in obesity in making the mice obese. Right? In this publication, so in that case, you get Megamonas as a very good candidate. If you can reduce, let's say, reduce the number of Megamonas in the population, right, you might be able to prevent obesity. All right, that's a good one. But when you look at the statistic here, all right, turn out that Megamonas are mostly found in China, all right, and very, very few in Europeans, which means this would be applicable only in China, not worldwide. If you want to take whatever intervention with Megamonas, it will work only in China. All right? That is one very big problem about microbiome because there are so much variables across country and region and nations. Okay, so when we look at the genes, microbiome things, so we actually focusing on the sequence, which is the amplicon sequencing, metagenomic or third generation at here. But you can see that not just genes, right? What we can tell the real effects for the gut microbiome actually is their function, which means that you have to look into beyond the dimension of the sequence, look into metabolomics, right? Metatranscriptomics, metaproteomics, or even single cell sequencing in the futures, all right? But in terms of clinical application, you can see that this sequencing are cheaper, all right, but it gets more and more expensive, all right? And accessibility is why sequencing are easiest one to do, but when you keep going to very sophisticated method, it's very complex, very difficult to do, and very hard to be practical in terms of clinics, right? So what we hope for now is that nowadays this kind of thing are big data, right? We have sequencing data and all these functional data, all right? Hopefully that if we have a big data enough, could we train AI in order to predict that once we get sequencing data and they predict actually the functions, all right? And in that case, we can use simple method like clinical tests to use to be practical in clinics. Okay, and for you, I would believe that if you become advisors for the team, then you have to get involved in some kind of medical education, right? In terms of education, we also have challenge, which means that now we have a new knowledge, right? New pathogenesis of disease, especially in NCD, which means that when people teach about diversity, you have to be involved with microbes. All right? When you talk about when the medical student learn about diabetes, then it have to be involved with microbes. Right? In the future, microbiology will involve every department. And more biochemical pathway to learn, more names to remember. All right? And at this moment, like I tell you, that we are not kind of like we are in the very early stage of learning about microbiomes, right? And for the application, it's still a long way to go. At this moment, there are no clinical certified application like tests to use in clinics, all right? But commercially, you can see that there are many companies providing gut microbiome tests to predict disease or predict the diet, all right? Or even like probiotics, that they claim to help to promote health. Imagine that our student once they graduate, there might be a chance that the patients get the test result from the company and take the test result to ask them that I get this test result, got microbiome result from this company, and they say that I'm going to get cancer. What you gonna do with it? All right. Do we have answers to prepare our students their futures? All right. So in that case, now in terms of microbiome, right, it's gonna change the basic knowledge of medicine, and we need to prepare for that. And it might not be the roles of microbiology department alone. Right? In the future, it might need to be integrated into multidisciplinary course or sections in medical educations. Okay, so last slide. So you can see, just to make it short, microbiomes is real. They have a lot of data supporting it, but because of the variation, all right, so everything are not so certain now that this gonna work, this population gonna work, but in this another population it might not work, all right? But hopefully that in the future, using the AI technologies and big data should make sense of it, all right? Thank you.
Thank you, Associate Professor Dr. Iyarit Thaipisuttikul, for your fascinating insight. I'm sure many of you have questions or comments. Let's begin our Q&A session. The floor is open.
Hi, Professor. So I was just wondering, like you said that like vagina has the ability to control like the proportionate of the microbiota, right? So I was wondering, I'm not sure if it's caused from the environment or anything else, but what if can we like take that advantage and put it into the other organs and then treat the dysbiosis?
So say that again. So you talk about the vaginal microbiota, right?
Yes,
And
so, which I'm not sure what causes that, because like it has like a similar proportionate in each other person, right? And I was just wondering if we could take that advantage to treat like dysbiosis in other person, like to control the similar ability as the vagina.
Okay, so actually this question have two parts. The first part is why vaginal composition are conserved, right? Meaning that they are kept the same profile for every humans. The second one for the application.
Yes.
Right? For the application, yes, there are many type of application. Even vaginal microbiome transplant, have been proposed to do that as well. Meaning that some people that have vaginal dysbiosis and very resistant to any other treatment. Those could be one of the treatment that can be used in the futures. All right, but the first part is why, right? Why vaginal microbiota are keep at the low diversities. Right? This is totally opposite from any other organs in human bodies. Right? So usually you can see that common sense, right? More diversity is better, right? If you have a forest with more diversity, it's better than forest with less diversity. But vagina is the totally opposite, right? Low diversity is the best one. And in terms of evolution, all right? Evolution meaning that if you have ecosystem and you let them go along the natures, right, from time, it will get more and more diversities. All right. If you want to keep the low diversity, for example, you're going to keep only one single organisms over time, then you have to put energy in it in order to preserve that. All right. Which means that in terms of evolution, humans have to put some energy in vagina in order to keep it that way. All right. Which means those low diversity of vaginal have to be very important for human evolution. For example, it might promote the health of the fetus or the healthy stage of pregnancy and so on, so that human can keep, keep diversifies and, not become extinct, for example. They are not just hypotheses, right? They are hypotheses, but they are still very fascinating. That's why vaginal microbiota have to be that to keep it at a low diversity like that.
Thank you. We appreciate all of your fascinating perspective and involvement. However, I'm sad to inform you that it's time we continue to our last topic. If you would like to ask further question,
May I ask one question?
Please go ahead.
SI-77. Yeah, just joined the party last night. I would like to ask about the difference between the vegan and general populations. And now, so the speakers told us that we have more names to remember, right? Yeah, it's very important because it's strain-specific for any disease, yes. So in terms of implement to the disease management, it's still difficult because the vitality of the microbiota and the strain-specific and the number. Yes, please answer my first question, the vegans and normal population.
Oh, yes. So for the population that who take vegans for vegetable base, the gut microbiomes have been shown to be very different from the meat base, right? But that is depend on the durations of taking as well. For example, if you just switch from meat base to vegetable tomorrow, right? Your gut microbiome might not be changed in a week. It takes time, right? So, but there's a study different from different cultures, like people in Europe who eat meat-based food, and compared to people in African, which is based on more vegetable and grains, right? Their microbiota are totally different. And usually gut microbiota in African people are favor more in promoting health than in Europeans. All right, but in terms of application to adjust the diet, that for sure have some application, but it take times to follow up how much it changed.
In case of implication for disease management, it's very difficult, as I mentioned. Can we use the product of the microbiota like a butyrate instead of the microbiota or not?
Oh yes, that's one point. This is called postbiotics, which means that's a product of microbiotas. All right. Actually, people start to study the postbiotic using different kind of short-chain fatty acids to try to supplement that into gut microbiotas, and then I believe that in early stage of the result, it gives a very good result for that.
So if you would like to ask further question or share some extra idea, no worries. There will be time for you after the end of the conference to do so. So thank you again for our professor. Now I will welcome our final speaker, Dr. Punn Augsornworawat, who will present on understanding and engineering biology to single-cell sequencing.
Hello? Okay, good. Everyone can hear me? Good. All right. So thank you for this opportunity today. I know what everyone's thinking. It's very cold, and so we should get out soon, right? So I won't take too much of your time. So we will go through briefly of what our technology is capable of and what it could do, and some of the things that we've done in the past that highlights our research. Right, so my topic here today is understanding and engineering biology one cell at a time, right? And we're going to go into a little deeper into this technology called single-cell RNA sequencing, right? And so my name is Punn Augsornworawat. I am from the Department of Immunology at Siriraj Hospital. So we're right here at the, just one building across on the 12th floor, right? And so to begin with, before we could dive into this technology, I just want to highlight that usually in biology, particularly ourselves, so as a human, we're composed of many different organs, as we all know. So let's look at one organ. So what is this organ? Anybody can guess? A pancreas, right? Pancreas. So a pancreas, as simple as it may seem, it's actually very complex, right? It is an organ, just like any other organs, your brain, your heart, your liver, etc., your skin. You might think of there's one cell type or two cell types, but usually in all different organs, these organs are very complex with heterogeneous cell types. And so not all the cells are the same, right? So if we zoom in into the pancreas, we look closer, you can see this thing, little ball-like tissues. These are called the pancreatic islets, for example. And the pancreatic islet has a role in secreting various types of hormones to regulate your blood in your system, right? But if you look closely, you can see that it might be one cell type, but there is actually a lot of different cell types. So one islet, if you zoom in on this little cluster here, there is about 10 at least different cell types. And so you can see that even the smallest component of an organ can be complex, all right? So next example, there's about 10 cell types. And not only is there many cell types, there's different cell states too. So if you're in development, you grow, you get older, you can have state A change to state B, change to state C. Or in differentiations, or in disease, you can have cells of various types, right? So a cell type can have different forms as well, which makes things even more complex, right? This is an example of a cellular differentiation where we have embryonic stem cells, and if you can convert that into a cardiomyocyte, it's not A to B typically, but a gradient of different cell states from one to another. And the other problem is that sometimes these things can change back and forth to its intermediate forms. And so we're dealing with things that are very complex, very different cell types, different cell states, different forms. And so things need to be dissected in very strong detail. And so we sometimes, when we do scientific research, we think of things that are usually unicellular, but really in reality, we live in a world of multicellular because things are very different. And so this is in reality. When we do scientific research, we mix everything together thinking that, it's just A or B, right? So if you look at a typical qPCR graph that you see in many papers, we see, say, for example, gene A. If we look at the control and then treat it, for example, we see the stats, we end it there, right? This assumes that there's only one cell type. But in reality, we have to imagine things that are a mix of different things. So imagine that your body is composed of different fruits, right? Different types of fruits. And so if we look at things at a single cell level, we have to account the different heterogeneity, the different cell types that we have in our system. And so this is where single cell omics comes in, right? So we take different cells and then we look at it one cell at a time, just like in this. This is a microfluidic representation of how things are looked at. And so in single cell RNA sequence technology, we could look at different aspects or different data. One of the things that is most common is looking at RNA. And with this technology, we could look at all the RNA expression, all gene expression profiles. We call that transcriptome, right? And we do that one cell at a time. So this is an example of a pancreatic islet that we just looked at earlier, right? We mentioned before that there's many cell types. So if we take this islet here and then put it into this machine to do single cell RNA sequencing, looking at transcriptome one cell at a time, we could see that there's actually many cell states, cell types, cell states, right? You have the alpha cells, the beta cells, the endothelial cells, delta cells. Each cell has a particular function, right? And there are things that we could do with it, right? If we have more of this information, one of the examples that we did was that we took the pancreatic islet, we put the islet in the beta cells, particular, which are responsible for regulating your glucose levels, have strong implications in diabetes. We put these cells and then looked at it at a single cell level, right? Put it into a type 2 diabetic microenvironment. You could see that we could observe different aspects of things, different details that we did not expect. For example, if we put it under a specific condition, we could see that there are more states, more cell types that arise in compared to the healthy control, right? This is one example. We could also look at different cell states of differentiation, where in our study here, we took pluripotent stem cells that Dr. Kasem took the pleasure of describing it already. These stem cells could be transformed into any cell types in our body. And Dr. Kasem talked about liver differentiations. In my case, we talked about pancreatic differentiations, where we can take stem cells, convert that into pancreatic islets that are capable of producing insulin. And so if you look at this, you could see, using single-cell RNA sequencing, that a control here from a healthy human and those that are from in vitro, the ones that we make in lab from stem cells, also have different cell types, cell states, and they're not actually the same, right? In particular, the beta cells, which are the insulin-producing cells capable of controlling your glucose, have different transcriptome profiles, right? So this is a simple heat map. Looks complex, but if you just look at it with your blank eye, you can see that the coloring and a pattern between these two types are very different, right? Highlighting that, if you look at a few genes at a whole mixture, it's very hard to see, but if you look at it at a single cell level, we could see those details, right? And so one of our studies, we highlight that by taking these beta cells that we make, in vitro here that we see, they're very different from the natural humanite, but if you transplant it, we can actually convert that into a human-like beta cell. And this technology cannot be done without this tool called single-cell RNA sequencing because of the heterogeneity we find in these transplants. Back into differentiation, we also see that there are various cell types, cell states, within the stem cell differentiating beta cells, right? One example is that you can see that this cluster here, the beta cells, have this other population that we see in this graph. You can see that there's a continuum of cell states by looking at the cluster that they form one giant clusters, and you can see that there's a gradient of cells gradually changing from EC cells to beta cells. Now, EC cells are not beta cells, they're actually intestinal cells. And so there's a big biological question, whether or not EC and beta cells are by transcriptome similar, right? And so we could use single-cell RNA sequencing to actually engineer and understand the mechanism of what is going on. In this study, we found that a transcription factor, which is also an epigenetic regulator, can influence intestinal cells and beta cell fate. Right, so we had this experiment done where we utilized single CRISPR-A activation, where we attach a guide RNA of CTCF, we put it in the cells, and then we were able to overexpress that protein. And so we can see here that by overexpression, you can have increased expression of CTCF. And what we found with this experiment is that by inducing CTCF expression in the beta cells, we turn it into intestinal cells. Right? And this data cannot be done or cannot be analyzed with pure mixed qPCR. It has to be done with single cell because of the heterogeneous nature of our samples. This is another example of the use of single cell technology where we actually obtain patient samples from a rare genetic disorder. This is called Wolfram syndrome, right here. And so it's a patient that develops diabetes without a really known reason. And when we looked at their sequencing profiles, we found out there is a mutation called the WFS1 gene right here. And so when the patient has this mutation, over time, when they get older, their pancreas or their beta cells starts to deteriorate, right? And so we took these samples here from different patients, and then we generate stem cells using induced pluripotent stem cell technologies, turning fibroblasts into iPSCs, and then we reprogram it into the beta cells that we were making previously. And we found that looking at these islets, there is a very disrupted morphology, right? Now, morphology doesn't really tell us much. So we went ahead and did single-cell RNA sequencing to see differences and what we could do to fix these problems. And so with this, we found that there is developmental problems, but we really don't know the details. And so we did that with single-cell RNA sequencing to see how heterogeneous the samples are and what cell types are showing in these islets. We found out that there is a lot of off-targets, a lot of EC cells, right, intestinal cells, and very little beta cells. And if we look at the beta cells, they have disruptive gene expression. And so in this experiment, we took these patient stem cells, beta cells here, and then we performed CRISPR-Cas9 of the WFS1 gene, because that's the mutation that leads to this disease. And then we corrected it with the proper gene, right? So now this cell line has the ability to express proper WFS1 gene. And then we took those and then redifferentiated into the beta cells. We redifferentiated. And then we performed single-cell RNA sequencing, again, to see the composition of the cells, of the islets, right? And turns out we found that there are drastic changes in cellular heterogeneity, right? And of importance, the beta cell, the composition of the beta cells, as you can see here, jumping from 11% to 43%, suggesting that there is a big implication on beta cell development in these patients. And looking at the beta cell genes, we see a recovery of the genes important for functioning to cure diabetes. Right? So in theory, we could use single cell not only to understand biology, but to also manipulate genes and to see if we could use it as a cure to things, right? Now looking at this, we have a lot of information. We have transcriptome profiles, a lot of gene expression. We could look at pathways as well, right, using the database that are out there, and then computational tools to see what pathways are different between the two cell types, right, and particularly in the beta cells, because that's the problem of the issue. And then we found that with the WFS1 mutation, there is a big difference in these stress-related pathways, right? So knowing the candidates, knowing the origins, the mechanism, right, the basic biology driving these differences, we went ahead and then did a screening of different drugs that would target these pathways. And then we had a few, right? We had a few that we had a hit. And so we took, so this is an example of an insulin secretion assay, right? So we put, the light blue is, so we took these islets, we put it into a solution, and then leave it for an hour. We measured the amount of insulin that came out of these beta cells. And then we stimulate it with high glucose levels, right? Just like how you're eating food, right? You have elevated blood glucose levels in your system. And so naturally, it would shoot out a lot of insulin. So that's what's done here. So in a healthy pancreatic islet, you could see that there is increased insulin secretion in response to glucose right here. Now that's in healthy, right? The baseline is at about 2,000 micro international units. In cases of a Wolfram syndrome patient with the mutation, we measure the insulin secretion. We can see that there is a drastic reduction in the amount of insulin secreted, right? Partially because of the beta cell gene that is disrupted, and also the composition of the beta cells that's very limited in these islets. But regardless, there is very limited function, right? You don't see a big increase in insulin secretion in response to glucose. So we took these drugs that our candidate drugs that we identified, and then we treated with these drugs, right, based on the knowledge we found using single-cell technology. And turns out that it does actually improve insulin secretion in these defective islets, suggesting that single-cell technology, it doesn't only bring us a lot of junk information. If we use it correctly, we could use it to identify different pathways, the sources, to find new treatment methods for this. This is one example, right? So, and stuff like this, we're trying to pursue in different contexts, particularly in diabetes. With there's different forms of diabetes, and so we're looking into that as well. And there's other diseases that have not been looked into with this technology, right? So I think that's pretty much it for my talks. If you have any questions, I just want to emphasize that that basic research does really play an important role in defining new medical treatments. And so I don't have a real conclusion. I think I concluded most of my talks already from different papers. But if you have any questions, please feel free to ask. Thank you.
Thank you, Dr. Punn Augsornworawat, for that insightful and inspiring presentation. The floor is now open for any questions or clarifications. Does anyone have any questions that they would like to ask?
Start first questions. อาจารย์ปันครับ so normally when we start with the iPS cells, the organoid that we got, it will equivalent to the fetals. And you mentioned that when you transplant into the adult mice, and it get more maturity. So how that happen? Anyone can explain?
Yeah, so that's a good question. So the maturation aspect of developmental biology is something that is not well understood, both in literature or in science we know today. We do know, so that's first. When we use stem cell technologies to make new organs or tissues, we make something that is represented as juvenile, right? So like a baby. But we can't really make something that's an adult just yet. And we don't really know why or how to do it. Why is it important? It's important because the adult functions better than the baby, right? So the goal for this, in this field, is to find a way to make it an adult. In one of our experiments, we transplanted these cells that are baby-like into a mouse, and then we observe it over time. We found that they do mature in the mouse, in vivo. And so something about the in vivo microenvironment matures these cells. I tried to look into single-cell data, and we found a few candidates that we believe are strongly relating to maturation, but we still fail to see that in vitro. So I think it's a complex mixture of different things, not just the environment. Maybe the epigenetic plays a role as well. So a lot of different factors.
Thank you so much ครับ Our student มีคำถามไหมครับ Okay, good. Can I throw the microphone?
Have many people mentioned about that the single cell analysis are very useful, right? I want to know that this method also have the limitation that nowadays that we have to the limitation of this method. Yeah.
Can you repeat the question?
The many people mentioned that the single cell analysis is very useful nowadays, right? So I want to know the limitation of this method. This is help.
Okay, the limitation?
Yes.
Okay, the question is, what is the limitation of single cell analysis? Well, first of all, it's very expensive to do, right? So every time we do single cell analysis, we have to plan carefully of what we want to look at. this isn't something we can just do qPCR and to see if it's happening or not. We actually need a good biological question or samples. And there has to be considerable differences before we perform the experiment. There are cost reduction methods, such as cell hashing, where we can pool samples together to do one experiment. But I think cost is still a limitation. The other limitation is the sequencing depth. If you want to look at genes that are not highly expressed, particularly transcription factors, this low expression might not appear in single cell. And I think the other one worth mentioning is that because you have transcriptome at a single cell level, you have a lot of data to deal with. And so doing this analysis on X cell doesn't really answer your problem, right? So you have to utilize bioinformatic tools and high computing facilities to perform this task.
Are there any other questions? Since we have a little bit of time left, Dr. Punn, do you have anything that you would like to add on? Okay, we have one more question.
Yep, thank you, Ajahn Punn, for such an amazing presentation. I have a question, sort of like adding to what Ajahn Kasem asked just now, something to do with like when you put it in vivo, the transcriptomic profile is sort of like different from in vitro. And based on my experience, one of the reasons for that is because, yeah, as you mentioned, it's because of the microenvironment that the cells are exposed to. And one of the reasons is because there are something to do with the mechanosensing pathways that could involve in terms of how the cells differentiate or choose its lineage and some things. So I just want to ask, like, based on your ongoing projects right now, or I don't know if you dig deeper into that, is there, do you have any ideas of how like the biophysics or like the mechanical force surrounding the cells affects these difference in transcriptomic profile when we put in vitro versus to in vivo?
Okay, also a very good question. We did an experiment a long time ago where we in parallel, we have one set of the cells that are floating, right? An organoid floating in the incubator without any mechanical effects. And those that were transplanted, right? And we have those same batch differentiate, we let it off at the same time. And then we did single cell analysis to look at the differences. Turns out that there are two factors. So one is time dependent. If you leave it enough time, it changes. But the key maturation factors do not go up as like those that are in vivo. And so that actually suggests that the microenvironment, the stiffness, the tissues, the 2D architecture might play an important role in this as well. There's also a strong evidence in some literature where we can put these cells into a 3D architecture with scaffolds that are made of matrigel or collagen and so forth. So ECM, right? And they have different gene expression. And so I think to answer your question, the 3D architecture and the mechanical stiffness, the microenvironment plays a big role in this.
Are there any more questions? Dr. Punn, is there anything you'd like to add on?
Okay, how much time do we have? I was afraid that I was using too much time, but I rushed on it.
Around 10 minutes.
Okay, we have 10 minutes. Yeah, so this is ongoing work that is something that people are interested because it has the potential to be something that could be applicable for regenerative medicine. So particularly, for example, what we call ATMP, right? This is something that the country actually is trying to push forward to, where we could develop new therapies to treat diseases that are never curable. Right? Diabetes is one of them, where it's a disease where there's cells in our body that degenerated or die, and with this issue, there really isn't something we can do about it, because once the cells are dead, it cannot regenerate in itself. And so we have to find an alternative method to make new sources for these cells. And so one of the things that people are trying to do in research in this field is try to find ways that we could make new organs, including Dr. Kasem, where he's making liver as well, make new organs so that one day it could be transplanted into patients. Although this is something that I think would take a long time before we get there because of the regulatory issues and then the QCs and the functional validations. This is something that we have to look at in the future. But it's definitely something of, I think, that should be valued in future research.
Okay, once again, I would like to thank Dr. Punn Augsornworawat. That brings us to the end of this session. As we conclude this session, I would like to extend my heartfelt gratitude to all of our guest speakers. Your expertise and knowledge have truly enriched our understanding of these groundbreaking topics.
I would also like to thank everyone in the audience for your active participation, thoughtful engagement, and enthusiasm. We hope you have gained valuable insight from this morning's session. If you have further comments or questions, please feel free to ask our speakers. The professor will be waiting at the front of the auditorium. You can also leave questions via the email that are provided on the screen.
In addition, please kindly complete the evaluation form provided on the screen so that we can use your feedback to improve our future events. We look forward to seeing you again for the upcoming activities.
Thank you and enjoy the rest of the event.